Imagine a world where predicting how a drug molecule behaves, its shape, stability, and interactions, is as fast as a classical force field, but as accurate as full quantum mechanics. That's no longer science fiction. Today, we're diving deep into AceFF-2, a next-generation machine learning interatomic potential that's redefining what's possible in molecular simulation, especially for drug discovery. Built on a foundation of over 2 million DFT-optimized small molecules, AceFF-2 isn't just another neural network potential. It's a precision-engineered force field trained to handle the full complexity of real-world drug-like compounds: carbon, nitrogen, oxygen, sulfur, phosphorus, halogens, even boron and silicon, all with explicit support for charged states, including highly charged species like phosphate groups. And unlike many models that sacrifice accuracy for speed, AceFF-2 delivers near-DFT level accuracy while running at speeds that make it viable for routine molecular dynamics workflows. So how does it work? At its core is TensorNet-2, a refined version of the original TensorNet architecture. The key innovation: a self-consistent charge prediction and neutral charge equilibration scheme, inspired by AIMnet-2, but adapted for maximum efficiency and accuracy. Here's how it works. During each message-passing layer, the model predicts partial charges from atomic features, not as physical charges, but as learned scalar embeddings. These are then adjusted via a neutral charge equilibration step, ensuring the total molecular charge remains conserved. The resulting charge estimates are used to compute long-range Coulomb interactions, which are combined with short-range, invariant energy terms. This dual-energy approach, local and global, gives AceFF-2 a powerful ability to capture both covalent bonding and electrostatic effects with high fidelity. But the real magic lies in equivariance. Unlike many models that use invariant architectures, which ignore rotational symmetry, TensorNet-2 is so, 3, equivariant, meaning it respects the physical laws of rotation while breaking reflection symmetry. This is crucial when simulating chiral molecules, where enantiomers can have drastically different biological activities. A model that treats mirror images differently, as they should, is essential for accurate drug modeling. Now let's talk performance. We tested AceFF-2 across a suite of rigorous benchmarks, from torsion scans to strain conformers, batched minimizations, and full molecular dynamics. In the Sellers et al. Torsion scan, which evaluates energy profiles across diverse molecular scaffolds, AceFF-2 ranks second only to OrbMol, and significantly outperforms on E2X, GFN2XTB, and even GAFF. Even more impressively, in the Behara et al. benchmark, which includes charged molecules, AceFF-2 maintains strong accuracy, unlike older models that fail catastrophically on charged systems. Then comes Wiggle 150, a test of strain energy prediction on 150 highly distorted conformations of adenosine, benzylpenicillin, and efavirenz. Here, AceFF-2 achieves a mean absolute error of just 1.76 kcal/mol, a massive improvement over AceFF-10's 2.73 kcal/mol, and far better than on E2X, GFN2XTB, or GAFF. And when we pushed it further, testing on larger, previously unseen ligands from the Schrödinger benchmark set, AceFF-2 showed exceptional generalization. While it struggled slightly with energy errors, due to size extrapolation, its force errors remained below 0.05 eV/A ring for the vast majority of cases, a gold standard for stable dynamics. In fact, when we ran molecular dynamics on a ligand bound to TYK2, using a hybrid MLIP-MM approach, AceFF-2 delivered 36.7 ns/day on an RTX 4090, stable, accurate, and fast enough for practical use. But speed isn't just about per-step time. It's about latency. For small systems, like drug molecules, latency dominates. That's why AceFF-2 leverages CUDA graphs via torch compile, enabling sub-millisecond inference times. In contrast, models like MACE-MPA-0 or standard AIMnet-2 are limited to 10 ms or more, a killer for small molecule simulations. And when we batched 100 conformers of imatinib into a single simulation, AceFF-2 delivered 90% faster throughput than serial runs, thanks to PyTorch's batching capabilities and CUDA graph optimization. Even more impressively, in batch geometry optimization, AceFF-2 scaled nearly linearly, making it ideal for high-throughput screening of conformational ensembles. Now let's address the elephant in the room. How does it compare to the state of the art? On E2X is fast, but limited to 7 elements and neutral molecules. It fails on charged species and larger systems. OrbMol is incredibly accurate and broadly applicable, but it's slow, and its model is not torch script compatible, making it hard to integrate into OpenMM. AIMnet-2 is powerful and equivariant, but still slower than AceFF-2 and struggles with charge extrapolation. MACE-MPA-0 is the slowest of all, not designed for biomolecules. AceFF-2 sits in a sweet spot. Speed comparable to AIMnet-2, accuracy approaching OrbMol, and full support for drug-relevant chemistry, including charged states, rare elements, and complex functional groups. It's not a trade-off. It's a breakthrough. And it's not just theoretical. The AceFF-2 model weights and inference code are available on Hugging Face, and we've provided full tutorials for integration into both ASC and OpenMML. You can run it today in your own workflows, no black box, no proprietary software. The implications are huge. From relative binding free energy calculations to lead optimization, metabolic stability prediction, and conformational sampling, AceFF-2 enables accurate, high-throughput simulations at a fraction of the cost of DFT. It's not replacing quantum chemistry, it's making it practical. And in the end, that's what matters: better science faster. So whether you're a computational chemist, a drug discovery scientist, or just someone fascinated by the future of molecular simulation, AceFF-2 is a milestone. A new standard. A new era. And the best part, it's already in your hands.